Thank you all for coming and welcome you to the inaugural Atlantic Stage International Speaker Series. So this is part of a national international speaker series for the stage program. And we're very excited because this is our first one that we're hosting here in Atlantic, Canada. Before we begin, I just want to acknowledge that Dalhousie operates on the unceded territories of the Mi'kmaq people. These sovereign nations had inherent rights as to the original peoples of these lands, and we pay our respects to them. Nova Scotia also has a rich history of the African Nova Scotian people, and we recognize their contributions to this land. It's my pleasure today to welcome our in-person audience as well as our national online audience to our session. For those of you that are online, you can put your questions in the chat during the talk, or at the end of the talk, there'll be an opportunity to raise your hand and we can welcome you that way. We have a lot of trainees and learners in the room, so I'm expecting very challenging questions from all of you in person. I've done that speaking, so I haven't introduced myself. I'm Svanja Stanojevic. I'm a respiratory epidemiologist in the Department of Community Health and Epidemiology, and together with Professor Mills Fleming, we run our Atlantic Stage program. For those of you that don't know, STAGE is an exciting training opportunity for students across Canada to expose trainees to high caliber international research. We facilitate interdisciplinary research, innovation, discovery, and learning, particularly around the statistical genetics and omics research, and training the next generation to make sense of all this wonderful data that's being collected. The International Speaker Series is a unique opportunity for interdisciplinary learning, and especially to bring our international speakers to Canada to help us. And STAGE promotes an extensive collaborative network. We have our very first, Melis, our first Atlantic Canada trainee, and if any of you are interested, I encourage you to visit the STAGE website to learn more. I just want to acknowledge Dalhousie University and Genome Atlantic, who have sponsored our Atlantic Canada speaker series. And I will kick off without further ado to introduce our invited speaker. So Dr. Louise Laurent is the co-director of the Center for Obstetrics and Gynecology Research Innovation in the Department of Obstetrics and Gynecology and Reproductive Sciences at the University of California, San Diego. And today she's going to share her research into how she maps the cellular composition and molecular programs and tissue architecture involved in placental development. She's going to share some of her insights into the single nucleus and high-resolution spatial multiomic mapping that she's collected from placental samples in her research practice. Many of us have gotten to know Dr. Laurent over the last 24 hours. She's been very generous with her time with our trainees and our local collaborators. And so without further ado, Dr. Laurent, we welcome you to Halifax in Nova Scotia.
Thanks so much. I'm so happy to be here and want to convey that, at least from my group and other people that I know, that we are very happy to collaborate despite what things might seem from the US side. And really welcome your questions, comments, if you want to get involved. What I'll be presenting is work from a project that's funded from the HubMap program, Human Biomolecular Atlas program, funded by the NIH. And as you will see, it was an opportunity to generate a lot of data. And I can speak to you more about the composition of the whole consortium. But so my group was a data generation group, or what we call a tissue mapping center. But we still have yet to dig in as much as we would like into the data. And so that's going to be our focus for the next year. I'll just be sharing some of the initial insights that we have come up with in our initial looks at the data. So so just to give a little bit of background to folks who maybe don't think about the placenta every single day of the year. Basically, this is an outline silhouette of a pregnant person. And so, here's the baby. This is the placenta. This is what we're going to be talking about the whole time. Just want to highlight that actually the placenta at term processes 50% of the fetal blood volume per minute and 10% of the maternal blood volume per minute. So this is why obstetrical hemorrhage is such a big clinical problem. So very interconnected between both the maternal and fetal units. And then zooming in a little bit, we're going to look at kind of the functional unit, which is basically a placental villus, which is this little branch-like structure. Fetal capillaries go in and out, and then it's surrounded by maternal blood actually here, which is fed by these spiral arterioles. Which then, of course, back up into the uterine circulatory system. And then zooming in even more, we can look at the different cell types. And I'm going to introduce them a little bit because I'm going to just say them as we go along. I don't want folks to get lost. So again, this is an intravillous space filled with maternal blood. Early in pregnancy, there are two almost continuous layers. The outer layer is called STB, or syncytiotrophoblasts. It's a syncytium. This is the major transport and hormone-producing cells of the placenta. And then underneath it is a layer of mononuclear cells called the cytotrophoblast, CTB. And these are the progenitor cells that give rise to both the syncytiotrophoblasts, or STB, as well as a third cell type, the extravillous trophoblast, or EVT, of which there are several subtypes. The EVT is responsible for anchoring the placenta to the maternal uterine wall, as well as replacing some of the cells in the maternal endothelium in the spiral arterioles, thus relaxing those arterioles and then allowing more blood to come into the intravillous space as pregnancy progresses. And then finally, what's not really shown here is it's the main immunomodulatory cell type that convinces the maternal immune system that the placenta and the baby are not foreign and therefore should be tolerated for the duration of the pregnancy. So I'll talk about these EVT, STB, and CTB through the talk. All right. So the way we kind of set up this project, I'm going to first talk about our term placental experiments. And basically, we enrolled 8 folks and collected placentas at term from 4 vaginal deliveries. So there was labor, 4 C-sections, 4 males, 4 females that were all divided in all those groups. And so just from the beginning, we wanted to do some sort of sanity checks. So we looked at differentially expressed genes between C-section and vaginal delivery and male and female fetuses. And you can see that there are some really strong signals in male and female. And this is because, of course, males have a chromosome that females don't. And so you can dig into this using, typical volcano-like plots. I know it's set on its side, but it is the same thing. And you can see like this really clear signal here. Again, this is Y chromosome. And then there's some, sex-specific ones. And then some really interesting set of things between vaginal and C-section. So this is great. People have done bulk sequencing forever. What can we do with single cell and spatial? So these are the limitations. One thing I do want to point out, actually, is that bulk sequencing, I think, still has a role, right? You can go much deeper on bulk than you can with single cell. You don't run into the issue of like Poisson distributions, where if you don't go deep enough, you just completely miss a transcript. So it's also a good way of looking at full-length transcripts. You can have very high-quality DNA that goes into bulk sequencing. So I wouldn't, completely not use it anymore. We still do bulk in conjunction with almost all of our experiments. And luckily now it's reasonably inexpensive, so it's not a real big investment. Of course, right? We have no information about the cell type. And also if you have a very rare cell type, you can miss its signal, of course. Right? Okay, so I'm going to move on. Now, I also want to talk about just really briefly that in our data set, we also did bulk matrixome profiling. So this is mass spec proteomics with the sample prep directed at extracellular matrix. We did this in conjunction with Alexandra Naba's group. She's an expert in this area, but basically just to emphasize, we are trying to build as much of a multi-omic data set across the same samples as possible. And according to Alexandra, this is very good data. So we did a good job collecting the samples, yay. And in this case, we wanted to ask some technical and biological questions. Some of these have implications for how we collect placentas for future studies. And then other ones are just sort of interesting things that distinguish the placenta from other tissues. So again, we had the same 8 donors. We looked at samples from what we called the maternal, the fetal, and the central aspect of the placenta, because the placenta has a thickness, right? And you might imagine there's different cell types in ECM that sort of contact the uterus versus the membranes around the fetus versus in the purely villus space. And in 2 regions in each placenta, sort of 2 biological replicates in each, we really didn't find that much variation. And so at least for ECM profiling, it appears that you don't have to really kill yourself and do 6 replicates per placenta to get reliable data. And this really has implications going forward, so that if you want to do further studies, then you can spend more of your replicates on true biological replicates of placentas from different individuals rather than doing replicates within an individual. We also looked at, differences between labored and unlabored. And again, we haven't dug into all of this as much as we would like. But basically, the placenta, unlike other tissues, because Alexandra Naba's group has done studies across multiple different tissues, what she pointed out is that we have unusually high levels of rare collagens. And then also things like elastin are notably absent in our data set. And then there's some interesting things like transglutaminase, which is may have implications in terms of the biomechanics of the placenta, especially in disease. Because we know that for certain placental diseases, the placentas are stiff. Okay. So I won't spend a lot of time on that because, as I said, we haven't really dug into our matrixome data as much as we would like. So I'm going to move on to single-nucleus studies. And so we use this technique called the 10x multiome, which simultaneously obtains both RNA and ATAC-seq from the same matched nuclei. And so you can see that the same basic cell types are identified. So these are 2 UMaps of the RNA data alone, the ATAC-seq data alone, and then the colors of the corresponding clusters are the same across both. And so there are some differences, but you can see globally we've identified and separated the same types of cell types, and then you can put them together to leverage both. And this is what we would consider our more definitive data set for our multiome. And if people want to talk, there are lots of considerations on generating high quality multiome data. This is actually our second try at it. We had basically a core make our first data set, and we found that the RNA-seq data was terrible. And so we had to redo this in our own lab, and we spent a lot of time optimizing the nucleus isolation protocol. And then as a reward for that, we were asked by HubMap to do a multiome on all the tissues for a trans consortium project. And I think it almost killed 3 people in my lab because we had a lot of time pressure. They had to generate all the data of like 20 different organs months or something. So if people haven't done it before, and single nucleus isolation is very tedious and painstaking. But we generated good data, so that should be coming out in a joint consortium publication, as well as this will be a separate publication from our group. But just to point out what we can find, of course, we find the cell types that we expect to find. So immune cells down here, endothelial cells over here, stromal cells over here. And then in this area are the trophoblast, and there are the subtypes that we talked about before. So there are the cytotrophoblast here. We actually have a subgroup of proliferating cytotrophoblasts that express cell cycle genes. And then we have a very small EVT group, which I think is somewhere buried in here. And then our syncytial trophoblast here. And remember, this is term placenta. And then we can actually see that there are some cells that kind of are pointing out, right, from the cytotrophoblast to the syncytial trophoblast. So they're likely differentiating or merging into the syncytial trophoblast here. But then there's also subtypes of syncytial trophoblast, and that's what we are actually digging into right now. So we were kind of disappointed not to see a lot of EVT, but remember the EVT largely is in the interface between the maternal and the fetal sides. There are some within the term placenta, but the other thing is that it's in a dense network of ECM as well. And so we think that this may be an artifact partly of how to get the nuclei out. In fact, in early studies, we tried to do single cell, and it was even worse. And that's why we thought, well, single nucleus not only would let us get the STB cells, but also access difficult to isolate nuclei, but that was only partially successful. But despite that, we have some really sort of interesting, and I'll come back to this because we've been able to crosswalk some of the data sets with this. And so going back to the STB, you can then identify the subclusters of STB and the particular transcripts that define different subclusters. So again, we haven't done as much as we want in mapping them to actual locations in the placenta, but at least we can identify some groups. Okay. And this is just one example of a particular transcript set in this cluster here. And this one does, obviously cluster separately on UMAP. And then this is another one here. It's a link RNA actually, which is interesting but difficult because a lot of links are not annotated in such a way that we know the function, but that defines this little group over here. And then Adam TS like one, maybe Christine knows, so the NMTS genes are associated with certain subtypes of preeclampsia. So this is a very interesting subset as well. All right. Okay, so going back, going to this technology and what's good and bad about it. So it does give a single nucleus resolution. In any given sample, you can see you can get thousands of nuclei. So you just saw there were 65,000 nuclei from 8 samples. This is essentially untargeted. It doesn't give you any spatial information, right? And then, as I mentioned, there can be some bias in capture of different cell types. It is kind of expensive still. So surprisingly so, like when you do the process, you're like, it's not that big a deal. That's the market, I guess. So that's why we only did 8 cases. All right. I'm going to move on to a spatial technology, the first one that we used in the lab. And I think this one's been given a little bit of short shrift. And partly it's because, one, you need to know a lot about your system to design an experiment. And the experiments are quite labor intensive. However, it's targeted, but it can detect 18,000 different human or mouse transcripts, depending on which flavor you get of this GeoMx technology. It's called GeoMx Digital Spatial Profiling. And if you're kind of good about it, you can look at very interesting cell types in different locations. And then once you get the data out, you have done what we call the segmentation already in the course of the data generation. So the data analysis is pretty straightforward. And maybe I'll define segmentation. So segmentation is basically the outlining, either physically or computationally, of the different compartments of cell types that you're interested in looking at. And I'll give you some examples here. Just want to make sure I'm not too far behind. Okay, so the way GeoMx goes, there's a lot of terminology. So what you first have to do is you take your tissue section, you stain it with the antibodies that you have decided are the right ones. You have 4 channels to play with. They recommend from the company to use, and this is Bruker, to use DNA for one and then 3 markers for the other. And so you have different colors here that you can use as your floors. Using what you see, you can define a region of interest, an ROI, and you draw it. It can be a circle, it can be a square. And you can see from ours, you can make it any polygon you want if you have enough patience. Once you do your ROI, then you can set up rules where you can define cell types within it that will be illuminated with the sort of capture illumination separately. So you can do basically like laser capture on steroids if you define things correctly, and you can set different thresholds for every one of your markers. And so I'm just using plus and minus, but we spent a ton of time defining the thresholds. And in that way, each ROI or region of interest, you can eliminate multiple and get multiple different areas of interest, AOIs, and each AOI becomes a library that you can then sequence separately, index separately, and look at separately. So in this way, my group ended up generating, I think, the most data sets, because every AOI is a data set in all of HubMap. And suddenly when we deposit some data, we had like 3,000 data sets. So basically, this is kind of the scheme that we used. So we used a DNA stain this is for the term placenta, pan-CK for all trophoblast, HLA-G for extracellular trophoblast, and CD31 for endothelial. And then what you can see is these are the rules we set up. And it turned out that actually HLA-G was not that useful because there are cells that are not quite mature EVT that don't express HLA-G. And so what we ended up doing is we defined EVT very similarly to STB, but they're in such distinct areas, and you can use cell morphology, that the ROI drawing was the way that we kept things separate. Yeah, so I'm going to go through. So for villus, so this is a villus area. You can see this really funny looking ROI. And the reason is that we even avoided areas where the microscope didn't capture a completely crisp image, because you can get erroneous segmentation if it's just a little blurry. But basically, we had kind of a squarish area, but we took out some of the blurry parts. And what's blinking on and off is the pan-CK primary, is our definition of the STB. And you can see this is term placenta. So the cytotrophoblast layer is extremely discontinuous. And so we don't really capture it separately. And then on the same ROI, you can use CD31 to define these beautiful capillaries, actually. And so we can capture those separately. That's a different ROI. And then everything else we just took as stromal immune. And so in this particular segmentation, we cannot separate stromal and immune, but we did get endothelial cells in STB, which is what kind of our focus was at that time in those. And then, as you can see, morphologically, the EVT are very different. And so we can just capture the EVT using pan-CK. And you can see that there is some HLA-G in some of the cells, but it's not completely uniform. So we just captured all of these guys. And then looking at endothelial cells, in this experiment, and I'll come back to this, we used the settings that Bruker recommends for a minimum number of cells and area to have enough to make a library. You'll see later that we actually pushed the limit on this, but here this is a large vessel, and we captured large vessel endothelial cells with the CD31 stain. And so you can put all that data together. And as I said, it's very trivial at the end of the day, because you just get a matrix of all of your data annotated. You know, if the person in your group, like my group, are very compulsive, each one is annotated as they pull the AOIs with what compartment they're from. And so you can see, you know, really obvious clustering of the different cell types. And then if you look at the vascular endothelial cells, you can even see that the large vessels and the capillary cells cluster very separately. And you can actually see markers that have been reported to be different in large and small vessels, and you can see those as well, very cleanly defined in your data. And then this is less well appreciated, but this is a transporter membrane protein that also is differentially expressed in the large and small vessels. And this one is actually really interesting because it is actually not only differentially expressed among different cell types. And one thing that's really important to know is, and this is why bulk can be a problem, a lot of different cells can express the same transcript, but they can be, in a given biological situation, only differentially expressed in one of them. So you can see that this transport protein is expressed in all these different cell types, but not really differentially expressed in vaginal C-section. But for the large vessels, definitely very strongly upregulated in vaginal versus C-section. So this is an example of how you can pick that apart using spatial. And this is another example of an aquaporin that's differentially expressed in labored and non-labored. And again, we can identify that for this one is it's in syncytial trophoblast where there's the differential expression of aquaporin with labor. Yeah, and then you can go back. I promised I'd come back to the multiome, and you can see that it is expressed sort of unevenly in the different clusters of STB, but definitely more in the vaginal than the C-section. So having sort of two different ways of confirming differential expression of a given target with labor in a given cell type. So we're going to move on to the EVT, which we focus with EVT on different locations. So what you can see here is this is the maternal side of the placenta here. Because it is a term, we didn't capture the full thickness on the slide. We've since worked with different platforms where we can actually get a full thickness. But here's EVT again. And then if you look over here, this is a different like septal area, and there's EVT there. So basal plate and septum. And then there are two other areas that we see EVT. There are these things called trophoblast islands kind of buried in the middle of the villus area. And then you can actually see them near the chorionic plate right under the membranes as well. So we captured those separately. And then we looked for differential expression of these different types. So we took, dozens from each of these compartments and then differential expression. And then what you can see is that the chorionic plate and island EVT populations are extremely similar. The basal plate and septal are extremely similar. And then the most different are the basal and the chorionic plate. So you can look now at the different subpopulations in different locations. And then you can look at, of course, what the different transcripts are that kind of distinguish these subpopulations. And so, so that was very enlightening for us. Some of you look at those genes, they're of interest because some of them are differentially expressed in preeclampsia and other placental disorders. So for GeoMx, the spatial information is preserved. There isn't cell type capture bias. Remember, we couldn't get those EVT from the multiome, but we could see them, and therefore we could profile them on GeoMx. And it's whole transcriptome. They are not single. It's not a single cell platform, however. And what we were, in the manufacturer's instructions, it says do not profile less than 20 cells. And as I said, it does take quite a bit of expertise to design your panel, validate all your antibodies, and then have somebody who is good and patient enough to do those segmentations. All right. So I'll talk now about a few imaging approaches. So we performed imaging mass cytometry, which is a spatial proteomics assay that uses antibodies that are linked to metals. And so we designed this 28-plex custom panel along with our collaborators at JAX. And this is just an example of, pretty pictures you can get looking at the chorionic plate area with markers like EGFR, which also mark the trophoblast, HLA-G. This is actually, HLA-G, which marks, of course, EVT, CD31, again, for endothelial cells, and then one of the collagens, which marks stroma. And just a really interesting little area that we're digging into more is that interface between the chorionic plate and the villi, where it looks like the villi are kind of like merging in, and you can see these different domains of endothelial and trophoblast cells kind of all there. So this is something that you can do. This brings up the issue of segmentation, however. What is a cell and what cell type is that cell? So we collaborated with a group at Duke, John Hickey's group. And so he developed this pipeline called Mesmer for segmenting cells and then clustering them. And there's various different approaches that I can go into the weeds with anybody who wants to. But basically, there are ones based on what are called nuclear expansion, detecting a nuclei using DNA staining, and then going a certain amount away until you hit another cell, calling that boundary cell. Of course, we all know that cells are not all round, and they don't have their border just because they hit another cell. And so that has some limitations. So John Hickey's group have incorporated protein markers that define, you can define like this is a cell membrane marker and then call that the boundary instead. But as you might imagine, this is still a very tricky part of any spatial technology. So anyway, so basically we only ended up generating data from 4 placental tissue samples. And I think we basically just killed the machine because they retired it right after. Apparently, it took a month to generate the data from 4 sections, and they had to replace the whatever it is that puts out the electrons. Can't remember what the part of the mass spec that's the most expensive, basically 4 times during that run. So beautiful data, but very expensive. And there is a new version of the imaging mass cytometer, which I've heard is much more robust. But anyway, beautiful data. These are 28 markers, and these are different clusters. And you can see that, you can basically do a hierarchical clustering and divide things by their expression profile. It becomes more interesting when you start looking at the annotation. This is the part that for us, as the data generators, takes us the most time because we are supposed to be the domain experts on the tissue. And so we need to look through the literature, our own knowledge, and actually go back and forth with the images too, to look at cell morphology, to annotate these clusters. And so not surprisingly, when we saw the multiome, there are multiple syncytial trophoblast clusters that we see. We can see immune cells, which I'll get to in a little bit, here and here, lots of endothelial cell clusters, which I'll talk about in just a second. So once we dig in, so this is where we went back into our images, and we noticed that some endothelial cells were Claudin 5 high and some were low. They were included in actually by one of our collaborators in this set of markers. So it really wasn't on our radar till we saw this really dramatic difference in the Claudin 5 expression you can see here and here in profiles that were otherwise quite similar. And when we looked, they were indeed vessels that were being outlined. And then it turns out that from other sources, there's just a couple papers talking about Claudin 5 in arteries and veins. And it turns out that arteries have much higher expression than veins. And then when you go back and you look at the actual signal in our IMC images, we can see like this is a reasonably large villus, that there are differences in very clear differences between the large vessels, some of which do express Claudin 5 and some of which don't at high levels. So now we can distinguish arteries and veins. And remember, so when we were doing the GeoMx, we actually would loop together multiple vessels together because they were small. I'll talk, so like this is what we did with GeoMx. And so we couldn't separate that signal for our GeoMx. We only saw it when we looked at the IMC. And then you can go crazy and then do really detailed annotation of different clusters. And our next step, which we're in the middle of, again, with the Hickey group, is generating what we call neighborhoods. So looking at what cells are next to other cell types and looking at the spatial relationships between them. But the annotation part is like all the hard work before you go into that. So moving on to, so that was proteomics, spatial proteomics. Now we're going to go to spatial transcriptomics. And we use the xenium assay. Some of you might have heard of the Visium assay, which had sort of a resolution that was multiple cells. We kind of skipped over that because I couldn't get my head around like how to deconvolute that data just conceptually. So I'm like, let's wait until the next technology comes out, because we knew that something higher resolution was coming. There's now a Xenium HD, which has like really small spots and can be agnostic about what you look at. But it turns out that when you, and Xenium HD is great because you can look at thousands of transcript types. Xenium kind of is annoying because you have to pick the transcripts that you want, at least when we did it. Now there's a higher plex, 5,000 transcript Xenium assay as well. The problem is this, that when you increase the number of transcripts you look at, your sensitivity for the technology goes way down. And so if you have rare transcripts, you will miss them if you use that agnostic approach. The other problem is if you have a weird cell type or a weird tissue like placenta, all of your genes of interest are missing from those big panels, which are mostly Israel-Onco. So in the end, it was kind of fortunate that we jumped and we did Xenium with a 300 plex panel that we put together. So yeah, so this is kind of boring, right? This is just a DAPI. This is where DNA is and the placenta, again, at term. But if you overlay the data from our 300 transcripts, then basically, this is just, I think, nuclear expansion segmentation with, you can ask things to pull back a little bit so you don't go to the very edge. This is what it looks like. And so there are all these sort of cell types that we can identify, again, very familiar ones that we've seen. A little blobby, I would say, because we're not using cell surface markers. And the other thing that we miss is, because it's nuclear segmentation, if you have a cell where in your section you miss the nucleus, you will miss the cell. And so capillaries often have these, the capillary endothelial cells are like, we call them sort of signet rings, right? I know that signet rings is for some cancer types, but I think this looks like a signet ring where the nucleus is here and then the cytoplasm goes around, but it's like a sleeve, right? And so many cuts, you're going to miss that. So this is one of the big weaknesses and the things that we are partially concerned about, because the vascular endothelial cells are these green things. And in the images of the staining, you'll see that you can see the beautiful capillaries, but they just look like little blobs when you do segmentation with nuclear expansion. This is just showing you that the thing about Xenium is that you can see where the different transcripts are being detected. And so, of course, this is an easy one, a little bit cheating. CYP19A1 is very highly expressed in the syncytial trophoblast. You can see it nicely outlining the villi here. All right. So what can we do? Looking at, this is differential expression done, so there aren't 300 things showing here, but then we can basically cluster and separate based on the expression of these different transcripts. Yeah, and one point here, this is basically like a little movie showing like what you can do in the software that comes with the 10X. And you can basically beautifully review for yourself the different cell types that you annotate. And we're just turning on and off the different transcript markers, defining, basically like our capillary or our vascular endothelial cells, some stromal cells, villus in the purple here. And we do a lot of this again. When we annotate cell types and clusters, we do a lot of this. So it's very sort of painstaking. One new technology that we're just sort of beta testing with Bruker is using their CosMx platform, which we haven't used a lot for transcriptomics, but they have developed a microRNA spatial approach, which is very tricky. As you might imagine, microRNAs are very short, and so it's very difficult to check them specifically, but it's basically showing, and I'm sorry, the lights are too bright. I don't know if we can turn off up there. Oh, nobody go to sleep.
That's okay. I'll look that up. Okay, I'll leave it off. Shouldn't have done that. We didn't. Sorry.
Yeah. So this is, of course, pseudo color here. This is concentrate and fine inside.
Little thing here. But this is a specific microRNA, and you can see that it is beautifully outlining some of these vessels. And again, we are a little bit limited from this beta test, we basically used what was off the shelf, which was an immuno-onco panel of microRNAs. So all our favorite ones for the placenta were not in there. But despite that, we can like find structures there. So this is, my lab has historically done a lot with microRNAs. And so we were very excited to see this, capability here. All right, so I am going to move on to actually what's kind of promised, which is looking at what we call our developmental cohort, which is placentas across gestation, which we generated a term data set, and then we started on the developmental. And so we're just completing the last data generation for the developmental set. And we redesigned our panel for GeoMx. So we decided we needed more channels. So we threw out the DNA channel because we didn't use it anyway. We still did keep the HLA-G because we thought in the early placenta, we might want to separate HLA-G high and low groups. And then we actually put two things into the FITC channel, CYP19A1, which is a syncytial trophoblast marker, and CD163, which is an immune marker. And the reason we were cool with doing that here is because, again, we do this ROI selection, and the regions that we expect, the immune cells and these STB are distinct. And then we kept the other three here. Okay, and so this is our segmentation strategy. I won't bore you with that. But basically what you can see here is that now we can separate, because in the first trimester, remember, the CTB is a continuous layer. We couldn't just use pan-CK. And so we use the CYP19A, and we can now beautifully separate that inner greenish CTB layer and the outer STB layer. And this is very useful for the first trimester and into the middle of the second trimester. And then actually this is showing that we can separate stromal, and did I already do the immune? I didn't note it, but we can tell the immune cells in here as well. They're kind of like an aqua color. The other thing also is, as I mentioned, we used to clump multiple vessels together the old way. And then now we do individual vessels. And what we did is we did a practice run, and then we showed that if we had at least an area of the AOI of like 1,100 microns, that we could get good data. And so, failed QC, passed QC. And the way we define this is that there are negative controls in the platform. So we have to make sure that all of our signal is above the negative control thresholds. So now we just do individual vessels. And so this is where we can separate the veins and artery AOIs. They cluster very separately and separately. All right, so then going back to our xenium assay, we used the same one as I showed before, and we collected samples across gestation. I believe there's almost 100 divided out through all of gestation. And I'm just giving 3 examples here to illustrate. First, second, and third trimester. You can see that the morphology of the tissue changes a lot in that time. So all of the cell types are color-coded the same. So STB in blue, so there is the STB everywhere. CTB in yellow, and you can see that it goes from a continuous layer in the first trimester to a discontinuous layer in the second to quite rare cells in the third. Pre-syncytial trophoblast, again, much more visible here in the first trimester than any of the others. And then EVT, truly in the third trimester, there are fewer. They are more localized in certain areas of the placenta, but in the first and second trimester, very prominent cell type there. And this is just doing bar graphs of quantification across many different sections. Again, just showing changes in the fraction, right, of the different cell types over gestation. So a lot of sanity checking that we do. And then going in and looking at the xenium markers that most clearly distinguish those 3 cell types. Those 4 cell types. Okay, so a lot of this is known, but you can now dig in and look at more subtypes. Right? So this is actually a full like first trimester conceptus, right? So just zooming in over here. If you then look at subcell types, you can see that there are three distinct types of CTB progenitor cells. There are actually distinct types of pre-syncytial trophoblast cells, and then four types that we can see of different extravillous trophoblast types. And you can see that they form kind of a gradient along the cell column. So this is where most of the, as you can see here, most of the conceptus is. So the uterine wall is out here. So going towards the maternal cells, you can see this gradient effect of progressive change in cellular transcriptional phenotype, at least. Yeah. And so then you can start looking at the things that distinguish between all of these cell subtypes as well. Yeah. Now, this is very early stuff. And again, this data just landed a few weeks ago. We wanted to look at quote unquote 3D. We kind of, the whole HubMap consortium calls it 2.5D because it's basically serial sections and then basically reconstructing some 3D aspects of the tissue by putting that together. So we selected 3 exemplar cases, one from each trimester. The G4X, I think it shows on a different one, has a very large format, and so you can get full thickness, even term placenta. But this is, for example, showing a early first trimester, I think about 5.5 weeks. So we can get the entire thing. And then we took 20 serial sections. This is a second trimester, yes. And it has both the maternal and fetal surfaces also. And then this is term, where again, we have maternal and fetal surfaces. We get the full thickness here. And we can arrange them actually all on 2 sections on a given flow cell. And then we did 10 flow cells, basically. Yeah, so I don't have like a lot there. We did a little bit of segmentation so far, but it is a new data type, so we have to work a lot more on that. Yeah, so basically, the other part is after segmentation, we're working with a bunch of other groups in HubMap for doing the reconstruction. Actually, two other groups, and they each are working together on different aspects of the reconstruction because there is both just registration, like what lines up between the sections, and then actually building the models of structures across and doing imputation of missing data. So for us, we were lucky. We actually tried to get 24 sections and the last 2 slides failed, but they were at the end. And so we don't have any gaps. But as you can imagine, in some of the other organ systems through HubMap, they've been trying to do the same thing, and they have dropouts in the middle where a given section folded or something like that. So that's why the imputation is really important to fill in those missing data. Anyway, so yeah, so I just wanted to thank a few people. I would say the person or the 2 people who have held everything together are Scott Lindsay Hewitt, who either generated a bunch of the data himself or coordinated it with other folks in my lab. Valentina, who is our lead clinical research coordinator and who either personally or supervised the collection of all of the samples. As you can imagine, trying to collect those seminal samples across gestation. We have 2 major clinical sites, and it just takes a lot of work. And so we actually developed much more rigorous sample collection protocols for this project, because if you're trying to look at transcripts in spatial, you need to have really optimized even preservation, storage, embedding, and sectioning. And then Monoprost is a pathologist, so she's instrumental in telling us what's where and whether. We did do some selection of cases to get rid of cases that had really weird abnormalities. Don't worry, we know what they are. They're still in the freezer because, of course, we do want to look at those abnormal placentas at some point, but this was more of a mapping exercise on normals. Paul leads the JAX group, so they did the IMC, the imaging mass cytometry, and the early multiome. His group is here. and here. And then John Hickey, I mentioned here, and then his master's student, Augie, they did the whole mesmer segmentation, and they're doing the neighborhood analysis now. And then we have some data folks on our side who help to basically keep track of all of our data. One thing I will say, because I think a lot of you guys do a lot of data work, that I've spoken to some folks, and I'm interested to hear more about how data is shared, stored, all that in Canada, because it is definitely a challenge, even among a given consortium. We have spent a lot of time with the data groups to develop metadata schema, and then even more time filling them out, and also babying them through the ingestion process. I started this as a joke, but I think it's actually true now that we spend more time uploading the data than generating it. And it's extremely painful. And I wish we had that time back to analyze the data. So I think it's a shared challenge for everybody, but in the end, right, if we really can generate interoperable data and share it, I think that would be a huge win, but it is extremely challenging, at least from a group that does sort of the data generation side. It's a huge lift. And looking back, we budgeted our grant completely backwards for getting these sorts of things done. So yeah, but happy to learn from anybody and happy to take questions. Thanks.
Thank you so much. The amount of work and to see the data generated. Maybe the Hugh VMAP, is that going to be publicly accessible one day, or is it kind of a closed consortium to begin with?
Yeah, I would love for people to dig into it, that we have data and other groups have data that are supposed to be publicly released already on the HubMap portal. So pitch for anybody to try to get at it. And like personally, I'd love to hear if you can access our data, and I'd be happy to give feedback. We initially had trouble ourselves getting it. And so we spent yet more time saying, look, we want to do this because we also want to look at our data in the context of other people's data in the consortium. So I think we figured out that internal system, but it is supposed to be public facing. So yeah, please let me know if you run into a roadblock, because I will escalate it, because we do want it to be used. And there should be already some that's out there.
Great. And you're talking about the placenta, but it's the whole bot, like you're getting tissue samples from...
Not us personally, but all the other groups. Like there's a couple kidney groups, a lung group, a skin group, healthy groups.
Great. Yeah. Maybe we'll start in the room. I see one hand up online. Finley, I knew you. Yeah, go for it.
I have a textbook data question and a biology question.
Yeah.
How did you go about developing textual data standards? You do this a lot with public health and international pathogen surveillance data. So how did you actually develop those metadata schemes?
Yes. So we tried to use, I'm going to forget the names, but like there's cell ontologies and molecular ontologies. I don't know if you know Kati Borner, but she does like the human cell atlas or whatever stuff. And so her group kind of outlined a lot of them. And then, yeah, I sort of blocked it from my mind, so we're not actively doing it right this session, but we've been through like 3 different versions of basically schema on placenta from the whole organ down to biomarkers of different cell types. It's called ASCT plus B table. The B is about markers. A is like anatomical structural tissue, something. And it's like giant spreadsheets that go from like describe every aspect of everything. And so where there are ontologies, they're brought into there. But as far as I know, some of this is sort of partly like Kati's brainchild. But yeah, that's partly, Valentina not only did that part, but she coordinated the input from all of our team, including Mana's group and my group and other placental biologists at UCSD to agree upon some of the terminology. So, yeah. In terms of the other schema, there's technical schema for like all the different, so every different platform has different technical schema, right? And so endless working groups that were built. So like the GeoMx one, which actually we generated the most of that data. So Scott did that. Xenium, which is multiple groups working on that. Now the next one right now is the genus like perfusion, on which some groups are bringing on board. And so there's a whole nother data set. Yeah.
Then we have a question online, and then we'll go back on audience. Shahiba, do you want to ask your question?
Sure. My name is Shahiba. I'm a postdoc at the University of Calgary, and I really enjoyed all the data that you had to share. Just as a note, I have tried accessing the placenta data on the HubMap, and I have not been successful. I have emailed the technical side because I wasn't able to figure out the annotations. So it isn't something that, at least to me, was immediately usable, but it might be a skill issue on my part. But the question that I have is that if you look within the last 5 years, especially, there have been numerous groups that have published their takes on single cell or single nuclear multiomic spatial profiling of the maternal fetal interface.
Yeah.
And when you look at the papers, everyone finds something a little bit different. And so how do we merge all this very disparate looking data of the same interface? And so, for example, there's like Wang et al. That had just been published in April, which had done a cross gestation with single nuclear and single nuclear ATAC-seq, paired across gestation along with multiomics profiling and proteomics profiling. So how do we reconcile all these different studies and bring them together to have like a true human placenta atlas that is congruent amongst different groups?
Yeah, you can just repeat the question.
Oh, yeah.
Didn't hear here, but
yeah. So one was that she was not able to access our data. So please email me and I'll try to connect you with the right people, both in my group and also across HubMap for that, because that should not be. There are some that are not released and some that are. So, but you should be able to get at least something. And the other question was that there have been a few papers recently. There was one from Susan Fisher's group that you were referencing in April on the maternal fetal interface. And there was at least some single cell ones from before. And that their takes on it were different to some extent, and how to merge all these. So I think that's part of the issue is when you look back at these, there's actually some, I think Mike D'Angelo from Stanford did one a couple, like 3-4 years ago or something. The thing is that everybody uses different platforms, or in the same platform, they use different panels of things. A lot of these earlier ones were on quite small numbers of proteins. I think the D'Angelo one was probably on the order of 20-ish, I believe, or maybe 30 proteins, and we're up to like 60. The other thing is sometimes when people use two different platforms, they don't have the same markers on both. And so we were really careful to put all of our protein markers in our Xenium and then add more so that we can do some direct comparisons between those things. So what we hope is that we've generated a comprehensive enough set that if we crosswalk ours and then incorporate these other data sets, we can bring some level of order and identify why potentially people are coming up with different cell types or cell subtypes. They talk a lot about different subtypes of EVT in these papers, right? And different models of how EVT actually migrates, right, and differentiates in the decidua. So yeah, I get what you're saying, but I think partly it's just because people have been picking at it with small numbers of markers and or small numbers of samples too, to some extent. The other part is, and this is where, I lean on my pathology colleague, Mona Parast. Some of these other studies, I don't think the Fisher one, but I think the D'Angelo one did things with tissue microarrays. And so it's very efficient, right, to do a tissue microarray because you basically take cores from multiple sections and then you embed them in an array. And so in one slide, you can do multiple tissues. We actually don't do that generally. We generally take a decent sized chunk, and sometimes we can put 2 of like a first trimester into one slice to be efficient, or in the singular, as I mentioned, we could put all three, but we basically get everything in there. The pathologists generally think, at least in the placental side, that if you take cores, you can miss a lot. And I get it, because if you take the wrong core, you will not get a trophoblast island, even though they're not rare in normal term placenta. And so partly it's kind of how you sample potentially and what you focus on that can get you there as well. I don't have like a perfect answer, but that's part of potentially technically some of the reasons people come up with different answers.
I want to thank you all for coming to our inaugural ISSS speaker series. There'll be more this year and in the coming years. And if you're interested in stage, please visit the website and see if there are opportunities that align with your training platform. And thanks so much for coming. And thank you, Dr. Laurent, for crossing across North America to join us. And it was certainly an enlightening and very engaging presentation. Thank you so much.